So we are now starting the recording part, so we can officially start with my colleague and friend Tommaso Pizzorusso co-chairing with me, and with Andrea Büttner is going to come a few minutes late. We are the three chairs of this roundtable about technology and science related to the development of and the implementation of the sensing possibilities, which is the main theme of this very interesting today meeting. So the idea is to have, we will have a lot of very interesting snapshots, short presentations about different topics, and I'm going to introduce the first speakers one after the other. Then I will give the floor to Tommaso and Andrea for the next 2/3 of the presentation. So the idea is, again, very quick presentation, and then at the end a quick discussion if possible, if time allows.
Yes, I just wanted to show very briefly a few suggestions from unicellular organisms about sensing. So the idea is to look at unicellular organisms for elementary sensing functions, and I will highlight just two. One is the sensing of confinement, which we can relate to touch, and another one is sensing of light, which enables these organisms to move towards light. Okay, so the organisms are small. They are 50 microns long and less than 10 microns across. Eugenia gracilis is the name. I mean, I call alga in inverted commas. And here you're seeing them swimming. So this is a combination of optical microscopy and then elaboration with a model that we have elaborated in order to understand the swimming behavior of this organism. It swims with distinctive helical trajectories, and I will say something more about it later. Now, if you put this organism into a tapered capillary, so in confined conditions, so that the confinement becomes more and more severe as the organism moves along the capillary, you will see a change of behavior. So from normal swimming, you start seeing that there are some doubts about swimming towards more confined areas, if you want. I mean, of course, this is a brainless organism, so I'm playing with this idea of having doubts. Here there is also the thought of going backwards. And as you will see, when the confinement becomes extreme, there is a definite change of behavior, and the organism goes backward towards regions of less confinement, and with a completely different motility strategy. So from swimming, which was the starting one, to something that is reminiscent of crawling of an earthworm. And so we are seeing here, obviously, that the organism is able to sense the presence of the walls and to start a different behavior. So it's a mechanism of sensing and response in an organism that has no brain. So it's all based on passive physical mechanism. And the other example I wanted to show is about sensing of light. So how does this organ sense light? So it's intrinsic into the swimming behavior with a beating flagellum, which is eccentric. The fact that the trajectories of the organism are helical, and the red spot that you see in the movie appearing and disappearing, now it's hidden, and now you see it. That is the photosensitive organ. And the key aspect that I'm trying to highlight is the fact that the motion behavior is swimming along a helical trajectory, and as the organism traces the trajectory, it rotates around the axis of the helix. And this is a consequence in the fact that the eye spot, the red dot that you see in picture A, is always faces the outward direction. And in this way, if light comes from a side, because of the presence of an opaque region, then the photosensitive organism is periodically in light and in the dark. While if light comes from the top in this picture, then the photosensitive organism is always lit. And so the basic response mechanism is that if the photosensitive organism feels a periodic signal, then this is a sign that there is no proper alignment with light, and the organism will change something in the beating strategy to align with the light. Okay, so these are the two major mechanisms I wanted to briefly discuss. And okay, so the idea is that we typically use these things to think about how to engineer shape-changing devices that can mimic the wonderful abilities that you find in nature, and to construct maybe you know, biomedical devices, steerable needles or artificial muscles. And of course, with these two suggestions about the possibility of feeling contact and light, you know, the frontier is to enable these tools with sensing devices and proprioception. So that's all what I wanted to say. I'm putting some references here for later later use, and maybe I stop sharing if I manage. I can switch on the camera to say hello, and I can return the field to the chair.
Thanks a lot, Antonio. Thanks a lot. Very interesting, and you're going to see in the next few minutes also many different applications and possibilities related to sensing. In fact, indeed, the next speaker is going to talk about something quite different in general. Francesco Greco, again from Scuola Sant'Anna, is an expert on material science and in particular soft technologies. And Francesco, the floor is yours. Different application, very interesting as well. Thanks a lot.
Thanks, Silvestro, for the kind introduction. So I selected for today a particular topic of my research that is about epidermal or tattoo sensors. And starting from this, I think a lot of you can probably knows about epidermal electronics. It's a form of electronics that is really not only flexible, but also stretchable, and that can be conformably adhered on the skin. There are several examples of these epidermal electronics. I selected here just two pictures from probably the pioneer of epidermal electronics, Professor Rogers. And essentially, you embed all the electronic components inside a soft, ultra-thin polymeric membrane and stick it to the skin. For doing what? For monitoring various health parameters. And this is the approach that we followed with my colleagues for developing ultra-conformable organic bioelectronics. Here, we try to follow the line of organic electronics, and in particular of printed organic electronics, to place electronic components, sensors, on top of temporary tattoos, essentially. So tattoos that can temporarily stick on your skin. We tried to develop the technology of printed electronics to enable, for example, these unperceivable skin contact electrodes. You can see in these pictures some example of application of these tattoos to detect various electrophysiology signals from the body, like the heart signals for electrocardiography, or muscle activation in electromyography, and the brain activity with electroencephalography. You can see in the figure on the left how thin this polymeric membrane is, and this is one of the reasons why these materials are so unperceivable and so and allow for such an intimate contact with the skin. I took this example because I think this is a very good example of a research field in sensing where you need mixed competencies in a group, in a research group. Of course, I'm a material scientist as a background, so of course you need competencies in material science, in physics and chemistry, and I named here a few subfields and some particular aspects that are important for the developing of these tattoo-like bio-monitoring devices. But of course, these need also competencies in the group, coming from bioengineering and medicine, especially for the biosignal analysis and interpretation, but also the skill structure, the biocompatibility, the comfort of the patient, which is another aspect which is very important. And then, of course, you need competencies in electronics and also in design and mechanical engineering in general, because you want this structure to really stay on the skin, to allow for mechanical deformation, and so on and so forth. So, from the in our group and in doing this research, we would like, of course, to have someone who has all these competencies, and this is very difficult, of course, to get. So I identified two key factors in this research. The first one that I already mentioned is the interdisciplinary crosstalk. So you need actors that can really be able to speak different languages and to learn new languages. And the second key factor, according to me, is hands-on experience. So instead from starting from a theoretical, let's say, or more low level, let's say, background, enabling the people working in this research with hands-on experience multiply the chances for this interdisciplinary.
Otherwise, we can go to the next speaker eventually, and then we are back to Francesco when he's back, if he is back. Okay, Calogero, okay for you?
Okay, fine, thank you.
Okay, so the next speaker is Francesco. Are you around? No. Okay, the next speaker is Claudio Oton from the other institute on working on telecommunication. From Scuola Sant'Anna, working on photonics. Claudio, are you around as well?
Yes, can you hear me?
Sure, excellent. So share your screen. We are going to have almost back-to-back two presentations on photonics. And the first one is Claudio. Please, you can start sharing your screens.
Well, hello everybody. My name is Claudio Oton. I work in the Institute of Mechanical Intelligence in Scuola Superiore Sant'Anna, Pisa. And first of all, I would like to thank Calogero for the invitation to show what our research activity. And he asked me not only to present the research we do, but also the skills that are needed to work in our field. So that's what I will try to do in these 5 minutes.
Thanks a lot.
So, I'll speak about two technologies. The first one is optical fiber sensing technology, which is a technology in which the optical fiber not only transmits the signal, but it's used as a sensor itself, because it can be made sensitive to temperature, strain, vibrations, pressure, etc. And there's many ways of doing it. One very common one is using fiber Bragg gratings, which can be inscribed in the fiber. There are periodic patterns that can be written on the fiber that they reflect back very narrow peaks in the spectrum, and the position of these peaks can be tracked, and they indicate changes in any of these parameters. And the good thing of this technology is that you can multiplex many of these along the same fiber. So one single wire can provide many sensing points. Which makes it very, very interesting. So as advantages, I wrote here, they are very small because the optical fiber is like a human hair. They are passive, no electromagnetic interference, and you can make distributed measurements with one single wire. Of course, this technology has many applications because this is quite unique. Here's one example in which we saw, for example, if you want to monitor a bridge, you can monitor in many different positions with one single wire. So that you can detect specific places where the problems are occurring in the bridge. But there's many other applications in many other fields, as you can see. The other technology we work is silicon photonic technology, in which we leverage silicon technology, which is the one used for electronics, but we use it for photonics. So on the surface of the chip, instead of drawing electronic circuits, we draw photonic circuits, which are waveguides that guide the light through the surface, and they do whatever we want to do. And that allows us to miniaturize many optical devices. This is an example of a commercial fiber optic sensor interrogator that is based on discrete components. What we're trying to do is to miniaturize this device into a few millimeter device by taking advantage of these technologies. That implies a reduction in cost, size, weight, and power consumption. And we're working on this field for more than 10 years. This just to show a few. So who are funding our research? It's a combination between public funding and private funding. So the private funding comes from companies from different industrial sectors, including transport, energy, automotive, and also the aerospace. And regarding the skills, so when we want to hire people to work in our group, the first thing we ask is a degree in engineering, physics, or material science. And then there's a set of skills that I wrote here. They're useful because none of them is required, but of course, the more of these you have, the better the chances that you will be hired, because these fields are all related to what we are doing. So we encounter in many of our projects, we have to tackle many things that are related to all these fields. Regarding the training initiatives in our institute, well, we have, it was already mentioned, we have a PhD program in emerging digital technologies. We have also a master program, which is called PIXNet, funded by the Erasmus Mundus program by Europe. It's called Photonic Integrated Circuits, Sensors and Networks. And then we also have launched a seasonal school called Photons at, which I have specific slide for it because the registration is still open, and it's on photonic technologies for sensing applications. And it covers not only optical fiber sensing technologies, but also imaging sensors and LiDAR sensors, and the registration is still open. Okay, so I'm done. I hope I was in time, and I'm available if there's any question.
Thanks a lot, Claudio. Thanks a lot. And Francesco is back, but I think he said that more or less he presented everything. But again, just to summarize, we're talking about very small unicellular organism to soft materials to photonics. And then, of course, the next speaker is another new, recently acquired faculty of Scuola Sant'Anna, Stefano Palagi. Stefano is working on medical robotics and soft robotic and small robots. Again, another possible application in some way connected to Antonio, but more on the robotics by robotics side. So Stefano, please go ahead and share your slides.
Hello, thank you for the introduction. And while I'm sharing my slides, I want to thank you, the organizers, for inviting me. I hope you can see my screen now. Mm-hmm. Okay, so yes, as you mentioned, this is very much related, but on the technological side, to what Professor De Simone presented a few minutes ago. And yeah, first of all, I want to introduce you to micro robots. That is the main topic of this very brief talk. So if you think of micro robots as very small robotic devices made of miniaturized components, then you're on the wrong side. So micro robots are very different from miniaturized robots. When we talk about micro robots, we talk about very microscopic stuff. And what you see here, for instance, is a large micro robot. It's something that is 1 millimeter in length. And well, this is already way smaller than what a miniaturized robot is, because miniaturized robots are usually in the centimeter range, let's say. And we also want these sub-millimeter devices to be untethered and mobile. Why? Because we want them eventually to move into the human body, to, for instance, deliver drugs in very specific locations, or do some treatments or some diagnosis inside the human body. So, first of all, we need to make them mobile. So the first question is how to make them move. This is not related to sensing, but just to give you an idea. These are two mobile micro robots that I developed throughout the years, and you see them moving and you say, okay, nice, they move. But what you don't see is that they're driven by external fields. And why is that? Because these micro robots, these mobile micro robots, are not self-contained. And we talk about field-driven mobile micro robots, and when we talk about them, we mean that the micro robots themselves are just tiny structures made of responsive materials, and most of the robotic functionality is actually off board. Here you can see electromagnetic coils that drive this very small magnetic micro robot. So in terms of sensing, the main problem you have is to track the micro robots. Especially if you think about biomedical applications. So usually you see in the papers that the tracking is made optical, and that's okay for lab experiments and okay for not too micro robots. And as you can see here, when you try to visualize 1 micron beads, even if you do it in very controlled lab settings, well, you are close to the resolution that you can get. And when you move inside the body, then you have, of course, limitations in terms of spatial and temporal resolution of medical images for tracking and controlling the micro robot. But once you move to microscale robots, that is, micro robots that may be self-contained and may have some sensing on board, the second problem is how can these micro robots perceive, I don't know, concentration of chemicals, temperature, light, check whether these have overcome some thresholds. Well, you might think, okay, I need onboard sensors and signal acquisition and signal processing, but you can have nothing of this on board. So whereas a normal robot usually have sensing, planning, and action and interacts with the world, when you design a micro robot, you cannot have the planning on board, at least. And what you have in the micro robot at maximum is some reactive system. And you can have these reactions by using stimuli responsive materials, for instance. And so you have to choose the right material for having the right reaction to the environment. And so the reaction is programmed in the material itself. And the third problem is the perception of gradients. This is something that is done usually by microorganisms. They might be chemotaxis, phototactic, and they can go towards light or a source of nutrients, or they can go away from them. And what they do is either they compare spatially for instance, the concentration of a chemical, or they do it temporally. By these strategies, they can, for instance, find nutrients in their environment. And if we want to do something like that in our micro robots, for instance, then again, you might think, okay, I need all these things, but you can add them. So what you can do is you can exploit some, for instance, interface microfluidic phenomena like diffusiophoresis. Here in this video, you should be able to see, even if the quality is not very good, what you see are very small particles that are moving towards a piece of crystal that is slowly dissolving, and they seem to move towards that as if they were trying to heat it, but these are just passive particles, and they are just collected there by chemical gradients. So this is just to let you appreciate the kind of phenomena that one can exploit in this field. So to summarize, if we want to develop microscale robots that have some sensing on board, we need, of course, to have engineering and robotic skills, but we also need skills in microbiology, because we need to understand how microorganisms, for instance, do these kind of things. Biomedicine, because also our applications are toward that field. But we also need strong skills in material science, in chemistry and physics. So you can understand how to develop these very, very small scale robots. You need a lot of skills. And well, I thank you for the attention, and I hope I was not too long.
Of course, after so many years, 2 years, I'm still not able to learn it. I'm muted, so thanks a lot, Stefano. Very interesting.
Thank you.
Again, another example of the complexity of sensing in different environments. And I'm very happy also to introduce the next speaker, Professor Antonella Bogoni. She's also working on photonics, and I'm very happy now again to congratulate her, because a few days ago she's been appointed full professor at the Scuola Sant'Anna. So Antonella, congratulations again, and the floor is yours.
Thank you very much. So can you see my screen?
Yes.
Yes, okay, so thank you. So I'm going to present the photonics integrated technology that we have in the school. It's an integrated technology for photonics that is an enabling technology for several applications. The original application is communication, is the reason of the big development of this technology in the last decades. But that can be also used for several sensing applications, like Claudio Ton already mentioned, in several fields like automotive, environmental monitoring, space, and so on. So in the school, we have a foundry for developing a photonic integrated circuits. The foundry is named Infotech. It's a 700 square clean rooms with different level of cleanness in the different areas of the clean room. It's a project that has been possible thanks to the support of the Tuscany region and donation of private industry. In the foundry, we can work with a 6-inch wafer, but we can also upgrade to 8-inch wafer. And the technologies that the Infotech can provide for prototyping in the research field, but also for small production for companies, are the silicon photonics, the glass on silicon, the hybrid integration. That means that we can integrate different devices realizing different material on the same substrate. And then we have a quite new technology that is the lithium niobate on insulator, and an advanced packaging pilot line. In particular, for the packaging pilot line, I want to spend 2 words because it's one of the capabilities that is quite rare to find. So it's very hard to find this kind of capability, not only in Italy, where I think Infotech is the unique center able to do that, but also in the European scenario. And we have in Infotech a complete packaging pilot line and for ground terrestrial application. But we are also upgrading the pilot line for space within a project in collaboration with the European Space Agency. These are a few examples of the kind of components that can be realized in Infotech, so you can see how they appear in order to be used in real application. But in particular, I want to mention that in the Sant'Anna environment, we have not only the capability to realize photonic integrated circuits, but we also have a background and an environment able to design these circuits and use these circuits in a real application. So we have a sort of a complete photonic fab that starts from the system requirements and go through the fabrication, packaging, and put on board of the PIC and real use of these photonic integrated circuits on applications. And I want just to conclude mentioning a few examples of kind of sensors that we developed in collaboration with Infotech. One of these is the photonic-based radar transceiver on chip. It is basically a chip that is able to generate and receive radar signals with a frequency agility that the conventional technology doesn't provide. And we also realized the optical beamforming network on chip that is useful for sensing, but also for communication. It's a sort of pointing of the beam used for radars for following the targets. That can provide a higher speed and a higher precision compared to the standard solution. And we realized also LiDARs in our fab. So it's a sort of optical sensor similar to a radar, but that exploit light, not RF signals, that can obtain very good resolutions compared to radar due to the intrinsic characteristic of light. And we are also working on onboard connectivity for developing more complex system on chip, where we need to connect different systems, but on chip. And we are working on a quite innovative material that is graphene that can increase the highest frequency that we the highest frequency operation that we can use for sensing, and can also improve the quality and the efficiency of our sensors. And finally, this is not sensor for now, it's just communication. We are working on quantum technology, in particular photonics quantum technology. For now, this activity is limited to communication, but it can also be extended to sensors using quantum radars or other kind of sensors. And this is a quite emerging topic in the state of the art. So this basically concludes the presentation. Thank you again.
Sant'Anna, thanks a lot. Thanks for the interesting presentation about the facility and the technology and the possibilities you can offer at the Infotech and in the Institute in general. And now I leave the floor to the other, one of the other two chairs, Tommaso Pizzorusso, who is going to follow up with the next speakers. Thanks a lot.
Thank you.
Thank you, Silvestro. So I will continue with the presentations, and so the next speaker should be Marco Cicchini from the NEST laboratory of the Scuola Normale. He's a CNR researcher at the institute that is embedded in the Scuola Normale laboratory. Please, I leave you the floor.
Okay, thank you, Tommaso, for the introduction. Can you hear me well?
Yes.
Okay, great. I share the screen. Okay, now the screen should be shared.
Yes, I can see. You can start.
Okay, so good morning, everyone, and it's a pleasure for me to give this short presentation in which I'm going to try to distillate some skills that would be required to effectively work in my lab. The main research focus of the lab is the development of portable platforms for molecular analysis. The objective is to perform an analytical analysis exploiting integrated sensor for detecting concentration variations or events. Sensors are generally based on several building blocks. The transducer is maybe the most important, since it must translate the observable, for example, a protein concentration in a plasma sample, in a measurable signal, which is generally an electrical or optical signal. Finally, the detector will read the signal and provide the output. As applications, so we are active in several fields. Among these, I want to cite the portable diagnostics for example, for brain pathologies or viral infection, environmental analysis for example, for bacterial contamination, food analysis, and industrial process monitoring in the framework of Industria 4.0. Surface acoustic waves are the enabling technology that characterize my lab. Those are mechanical excitations which propagate along the surface of an elastic material. They can be excited and detected by electronics devices, and since their energy is very confined at the substrate surface, they are very sensitive to surface modifications, such as the addition of mass. For these reasons, those can be used as mass sensors with a number of advantages with respect to commercial sensors based on bulk acoustic waves, such as the quartz crystal microbalance. Other technologies or methods that we develop and use are these ones. First of all, we exploit microfluidics for sample manipulation and preparation. Then surface chemical functionalizations for making the sensor specific for the analytes of interest. And finally, microelectronics methods are also employed for final measurements. In this slide, I show you an example of a workflow for a typical project in this field. For example, in Violoc, we must develop a portable architecture for monitoring the production processes of oil and wine. In particular, the system should be able to quantify polyphenols during mass fermentation or during the olive pressing process. The work is divided in 4 activities. So sensor development. Sensors will have to be designed, simulated, nano-microfabricated, and characterized. Competencies in solid-state physics, electrical, nanotechnology engineering are required. Microfluidic chips will have to be designed, simulated, fabricated, and characterized for their fluid dynamics properties. Competencies in engineering, physics, and material science are required. Chemical functionalizations will have to be performed. The right probe linker will have to be selected and possibly chemically modified. The produced surfaces, of course, will have to be characterized, and competencies in chemistry, mainly in chemistry, material science, but also in biotechnologies are recommended. Finally, biology validations will have to be carried out. This includes biological sample preparation, characterization, and electrical measurements and advanced data analysis. Competencies in biology, biotechnology, and chemistry are required, but also in physics and engineering. Therefore, what I'm looking for in a candidate for joining the Violoc research group? Well, 2 profiles would be well suited. One with strong physics or electric engineering background, but say open and with some previous exposition to biology and chemistry methods. This researcher would be dedicated mostly to technology development. The other profile would be with strong chemistry or chemical engineering background with some biological and electrical engineering skill. For example, with experience in electrical characterization of devices and or biological sample handling. This researcher would be mostly dedicated to chemical functionalizations and biological sample preparation. Finally, of course, all the researchers of the group will be involved in the complete device testing and data analysis. I think I stop here. I hope to have given you an overview of what is required for my lab. Thank you for your attention.
Thank you, Marco. Very interesting, and of course we have to discuss later in the roundtable about how to implement these students with these competences. But, okay, so we can move to the next speaker, who is Patrick Gadoros from the from Hungary, Department of Atomic Physics and Faculty of Natural Science.
Good afternoon, everybody. I'm Patrick Gadoros from the Budapest University of Technology and Economics. And I have prepared a couple of slides about a specific project aiming the development of an instrument, a small instrument family, based on fluorescent detection of water pollutants. I'm going to share a couple of slides. So the aim was the development of a modular directed immunofluorescency-based detector and instrument family for in situ complex water and water quality monitoring and its application studies. And among others, it included the expansion of fluorescence-based detection methods to measure major water quality parameters, development of intelligent sensors for modular embedded computing environments to detect given water pollutants, and the development of an information technology system to process data from the intelligent sensors, and of course the application of this instrument for various purposes and in various fields. For details, you can visit the homepage of this specific project written here in this slide. I have chosen, okay, there are many pollutants in water that can be measured via fluorescence, and in most living waters, there are algae. And I have chosen chlorophyll fluorescence because it's the most exciting of all. However, toxins are also very interesting. To be able to handle this issue, it is necessary to master skills from the fields of physics, chemistry, biology, and engineering. Of course, in practice, most problems arise in the field of engineering, detection and processing of fluorescent signals. So optical and electrical engineering is necessary. And of course, the applications are of various fields. Beyond base research, it is useful for industry, for example, wastewater treatment, environment protection, monitoring of surface waters, and of course, agriculture. Chlorophyll. So photosynthesis is most probably one of the well-known biochemical processes that exist. However, fluorescence of chlorophyll is a little bit less well-known. By the way, it's very strongly related to the photosynthesis. The portion of the absorbed light that does not heat the plant and does not used for biochemical processes is emitted by fluorescence, and chlorophyll has a very well detectable red fluorescent light. In fact, chlorophyll is an optimal agent for fluorescent detection since it has a strong signal and it is very easy to excite. The outcome of this project was a portable measurement system for this complex analysis. It consisted of four measurement modules and an integrated system to handle these measurements. One module was for the algae detection, quantification and specification. The other was for the measurement of total organic carbon content of the water. The third module was for the measurement of polyaromatic hydrocarbons, which are quite dangerous water pollutants. And the fourth one was the immunoassay fluorescence module for various toxins, mostly myotoxins and mycotoxins. And it was quite a conventional research and development project involving research groups from multiple universities and companies. But since at the universities there are quite strong involvement of students in every project, also in this, also to this project, did our students contribute in a very valuable way. Actually, MSc and PhD thesis works are in progress related to this project. MSc thesis works are in the field of physics and applied physics at the University of Technology, and PhD thesis works are at the University for Agriculture related to biology and biochemistry. Thank you for the attention.
Thank you, Patrick. And we can now move to the second part of the roundtable, the senses, body, brain, mind sciences. And the first speaker of this session is Professor Antonino Cattaneo from the Scuola Normale Superiore. Antonino is full professor of physiology and also head of the bio-SNS laboratory. So Antonino, please. Which is Alessandro Cellerino, again from the bio-SNS. He's associate professor at the Scuola Normale. Please, Alessandro.
Okay, fine. So, good morning, everybody. So, in my very brief talk, I will speak about two different things. The first one will be a completely different kind of sensors as opposed to what you have seen so far. And these are genetically encoded sensors. It means that some proteins normally with fluorescence, intrinsic fluorescence, are modified in order to report for some specific biological activity. So what you see in this slide is an embryo. The embryo is this one. And there are two fluorescent proteins extracted from coral species. And the red protein reports or shows cells that are not synthesizing DNA, so they are not dividing. And the green protein will highlight those cells that start dividing and therefore synthesizing DNA. So you see that there are, at this point in time, there are only red cells, and then you will see a burst of cell division, then cells become green. And while the cell becomes green, the embryo is growing in size, in width. And then they stop dividing, and then they will turn red again. So this is just one of the many applications of genetically encoded sensors. And I'm sure you will hear more from Antonino Cattaneo's talk. And for the rest of this very brief talk, what I will actually talk about is more generally aging of sensory systems. So this is something that is very pervasive in our life, but it's something that we do not really often give enough attention. and it's certainly, I think, matter of inspiration for engineering. And let's start with the most important of our senses, vision. So what you have here is the accommodation power, the change in accommodation power as a function of age. And you see that for the first 30 years of your life, the accommodation power is almost 8 dioptries. But then very soon, in the 40s, the accommodation power drops dramatically, and then, you know, depending on what age you are, you know very well what I'm talking about, or not yet, to the point that it drops to 0. And this is the reason why it is unavoidable at some point of your life to either have reading glasses or change your glasses. Similarly, also hearing is a subject to a quite strong loss of function. And this is due by cell death in the inner ear. So the sensory cells of the inner ear starts dying. And as a result of this, there is a very progressive loss of hearing that, however, is not homogeneous for the different frequencies. Now, what you have in this graph in blue is the frequency spectrum of human speech. And this red line here is the volume of a normal speech. So what you see is that you quickly start to lose very high frequency, but this is something that you hardly notice. You will start noticing that you have hearing problems when this loss of high frequency will impact into the frequency spectrum of speech. But what is very interesting is that those changes are not limited to the most peripheral part of our sensory system. So this has been studied in quite some details, again, in the hearing system. So the perception of sound. And briefly, what is highlighted here are regions of the brain that are activated when you hear a speech with white noise on top of it. And now what happens that these regions are not activated in young people unless you superimpose white noise on top of it, but they are activated in old people, demonstrating one very important aspect of the aging brain is that the aging brain tries to somehow compensate peripheral sensory loss with rearrangement or different activation of brain regions. And I will just close by saying what are the competencies in my research group. So the topic of my research group is the biology of aging, and people in my group typically have one of these 3 competencies. Either they come from data analysis of bioinformatics to analyze especially gene expression data, omics data, or from molecular biology transgenesis for generation of animal models that may express, for example, the specific sensors, or they have competencies in the study of the entire animal, so physiology and anatomy. Okay, that was all, and many thanks for your attention.
Thank you, Alessandro, for putting aging in the equation. And should we try again to present your slide, Antonino, or now I receive your slide, or should I go straight?
You go.
Okay.
There is no reason why I should succeed now.
Okay.
I will go quickly through. So I'll tell you the next one, please. So I will introduce one aspect about molecular sensors. And first, regarding teaching, I will say that I will remind you that molecular sensors are a central topic of a field that is known as synthetic biology, which in the context of ELISA is particularly relevant because synthetic biology aims at studying and streamlining the design of biological systems using engineering design principles. And in fact, at Scuola Normale, in addition, we are neuroscientists, but in addition to the neuroscience courses, I have been teaching a synthetic biology course since 2012, and we have been hosting and co-organizing a summer school an international summer school in synthetic biology. So this is something that I think would be important for the EELISA network. It is a community synthetic biology that can contribute a lot and benefit a lot from so in a bidirectional interaction. In terms of research, many topics are happening at BioSNS around sensors. I will only, some of which have been mentioned by Tommaso, for instance, sensing neural and synaptic activity, working with genetic encoded sensors, as also Sandro Celerino mentioned. An important activity in the lab is developing sensors for protein misfolding in living neurons in relation to neurodegeneration. But for the sake of time, I would like to introduce an actor here, is antibodies. This is a strong point of the lab, and we work on synthetic antibody technologies and synthetic immune system, and I will take you through what we are doing because it is relevant for molecular sensors. The next one, e quella dopo ancora. So in any molecular sensor, the binding event, the recognition event is a very important aspect, and the quality of the binding moiety is not at all a sinecura, and biology, the biologic side of the recognition, is not a commodity. So there is a need for high quality binding moiety, and antibodies are the ultimate natural and unsurpassed form of binders. When I say antibodies, we say all forms of engineered antibodies up to the very small single domain or nanobodies. And then, of course, the binding moiety can be coupled to transduction, signaling, actuator, and physical devices, but I will be talking about binding. Next one, please. So our interest goes back to ages ago when we pioneered the concept of using antibodies as genes, and this is going to be relevant. We normally use antibodies as proteins, but knowing that antibodies can be used as genes opens a whole new dimension to what we can do with antibody domains, for instance, subcellular targeting and etc. But this is relevant for something that I will going to tell you in a while. Next one, please. So one particular problem that today is really a huge problem in biology is the proteome complexity. And whereas the number of coded genes is in the order of 25,000, the protein diversity is orders of magnitude greater because of post-translational modifications, conformation, etc. So one problem is how to identify and sense proteoforms. These are called proteoforms. La prossima. And so, of course, proteoforms contain targets for diseases that are very difficult to target with drugs and very difficult to validate. And this is just a summary slide of showing how proteoforms are involved in disease. La prossima. And so what we have been doing is using a synthetic immune system in vitro, which is a library of antibodies in yeast cells that encompasses the whole diversity of the human immune system. And we have developed a way how to use these libraries in which we can select antibodies directly from the gene sequences with no manipulation of the protein. So the cell codes the antigen for us. And in a recent development, we also have been able to have the cell install the desired post-translational modification, such as acetylation or phosphorylation, in the target antigen, and in the same time to select the antibody against that particular post-translation modification. We call this PISA, so post-translational intracellular silencing antibodies. La prossima. And this opens the possibility and is opening the possibility of doing something that gene-based technologies cannot do, interfere directly with post-translation modified targets, and so such as the same protein coded by the same gene that harbors one PTM or the other could be and can be selectively targeted with these PISA antibodies. La prossima. So this is linking to a nascent project international that is the Human Proteoform project that has just been launched, and our technology is being presented to this Human Proteoform project because in all forms of the Human Proteoform project, the development of protein affinity reagents, as you see in this slide, is a crucial element of the Human Proteoform Atlas. La prossima. And so we are working on developing an antibody factor infrastructure in our lab. We have joined the Genome Right Project Consortium, which is another international initiative. We are the founding members of Genome Right Project in Italy, and our technology has been highlighted as a key European innovation in the context of the Human Brain Project. So I think that this antibody factory infrastructure could really be deliver a next generation reagents for what has been called the dark matter of biology, the post-translational modification targets that are a very rich source of disease targets for diagnostic and therapeutic purposes, but no FDA approved drug currently targets any post-translationally modified protein directly, exactly for the difficulties that I told you. So we think that this infrastructure could be really a resource for the community in Pisa, in Italy, and hopefully internationally. And just to conclude, there is another line of la prossima, per favore. Another line of work in the lab. I just have 2 slides on this. To use molecular sensors to target misfolded proteins and to image misfolded proteins in neurodegenerative diseases, such as Alzheimer's, that is a main interest in the lab. And I give only one example that is in the next slide. Se fai un po' di click, grazie, falli tutti così. No, vabbè, non proprio. So we developed sensors for a number of misfolded proteins. This is an example of tau. We have alpha-synuclein. We have amyloid oligomers. And the idea is to have genetically encoded sensors to monitor the progression of misfolding inside the living neuron in the context of several disease insults. And thank you for your time. And I hope that this can be useful to further discussions. Grazie.
Okay, so we move to Michela Tosetti, and she is the she's the director of the Laboratory of Medical Physics and Magnetic Resonance at the Stella Maris Foundation here in Pisa. And please, Michela.
So can you see my screen?
Yes, we can, yeah.
So I use my old, thank you, Tommaso, thank you, Calogero, to invite me. I just wanted to give to all of you a brief idea, or what called that I would like to have from ELISA and from Scuola Normale, Superiore and Sant'Anna. So this is a very old slide because this is what is my job, to study our 1,000 kilometers of neuron through MR technique. So it can make a lot of interesting things using different methods. Our job is to try to measure an invisible change with MR in human brain. And we apply this method, different approach, either pathology, to see physiological and biochemical characteristics of the brain related to physiopathology in clinical research, as well, as Tommaso knows very well, in neuroscience, understanding complex mechanism or age development and aging brain plasticity. But what we need to do that? So we need an MR scientist. An MR scientist that is not, as you say, related to a specific discipline. And I just rephrase our Galileo Galilei phrase. So MR scientists in neuroscience try to measure what can be measured and makes measurable what cannot be measured. And how to do that? We modeling physiological response that could be measured with an interaction of MR radiation with matter. We're looking for new R&D solution that can be detectors of signal as well sensing devices. We are. We design pulse sequences to excite the spin inside the brain, and after we modeling the RMS signal for quantitative measurement. That is the flowchart of what MR scientists should know to work in neuroscience. And MR scanner is a scientific instrument, so we have to increase the specificity and sensitivity through modeling method sequences, but also new technology. And so I want just to show an example. This is the response on a group analysis on sensory motor pathway. Normally MR is used in a group level, and for example with 7 Tesla that we have installed in Pisa, we can go to a single subject level, and moreover with the very precise sensory selective stimuli, differentiate between in blue and in yellow the single finger, and moreover the differentiation between sensory area and motor area. Why is it important, for example, in this aspect, that you can use here this to understand better what happened to the brain in congenital unilateral brain lesion. As Giovanni today showed, we can detect differences between normal and patient and try to figure out the response for a clinical trial in a rehabilitation process. In Pisa, we have the 7 Tesla MR system. It is the only one installed in Italy up to now. And as you see in the list of my lab, there is together physicists engineering. So I hire either physicists, either engineering, and all together work in method, development, safety, new nuclei, and so on. So I want to add to this phrase another one that is my second favorite one, and from Dyson. They say that there is a two approach, as in my lab. A concept-driven revolution that is typical of physics, and the tool-driven revolution that is typical of engineer. Exactly what I would like to have, with the help of Sant'Anna and Scuola Normale Superiore and the ELISA program, is an MR scientist of the future. I think about a master with the physicists from Scuola Normale that is a concept-driven science, from Sant'Anna that is more applied engineering stuff. So that is my need, if you can. Thank you so much for your attention.
Thank you. This was a very inspirational presentation, and I see many clapping hands.
Oh, thank you so much. And I can destroy my presentation. You.
We can move to our next speaker. I don't know if Andrea wants to introduce Tim. Is Andrea okay? She's here. I don't know if you want to introduce Tim.
I just switched over to Tim because as one of the typical new work situations, I'm struggling here. I think our Italian friends will be completely happy to hear what I'm doing right now in parallel. I prepared spaghetti. Spaghetti for my family because I'm in the home office. Sorry for that.
Okay, no problem.
But I would like to hand over to Tim, but I'm listening carefully.
Okay. So, Tim, go ahead.
Hi, everybody. My name is Tim Ward. I'm an assistant professor for perceptual psychology at the FAU, and I want to share my screen shortly. Can you see it? Yeah, and I want to thanks for the possibility to present some of my research here. And I want to, in my very brief talk, want to just give you a rough idea of the research which we are doing in our lab, and also give you some idea which I think are very important competencies and skills in this research field, actually. So we are investigating questions of human perceptual psychology. So we ask questions like, for example, how actually do humans perceive an apple when they eat an apple? So we usually have like an approach where we want to bridge different levels of analysis. So we have different stimuli. For example, an apple, just as a toy example, emits light, but also has volatile molecules and some pressure in the mouth and so on, which we can sense in our different sensory systems, like the eyes, the ears, and so on, and is then transduced to neuronal signals and processed in the brain. And at some point, somehow these independent stimuli and the independent processing is then combined into a holistic multisensory perception of, for example, a sour, fruity, and red and crispy apple. And the main idea and question in my research is actually, how does it work? How are the senses combining these diverse different signals, and how can we understand the different levels in which we describe this phenomenon? So the physical, the meta level, the neuronal level, what the brain is doing, and then the perceptual level. And to bridge these levels, you have several methodological approaches. Namely, we use psychophysics. That means we try to very precisely define auditory, visual, or chemical stimuli. And then on the other end of the processing and the psychological level, we want to define and measure very precisely what the humans are perceiving. For this, for example, use computational models, mathematical models, which, for example, describe a fundamental problem of multisensory perception, namely, how does the brain actually know that two stimuli are really coming from a common object and can therefore be integrated, or whether they come from different objects and therefore should, of course, not be integrated. And when we have described the perceptual level precisely, we also want to look and understand what the brain is actually doing. And for this, we use neuroimaging and neurostimulation. That means we can, for example, use estimates and parameters of the computational models, which we fitted to behavioral perceptual data, and link this estimates and parameters to fMRI data, where we measure in the brain multivariate signals actually, or also to EEG data, electroencephalographic data. And so we can characterize actually quite precisely where in the brain with fMRI and when in the brain, for example, independent auditory and visual signals are processed and then at some point in the hierarchy of the brain are integrated or segregated, depends on the specific stimuli. And yeah, by using this approach, I think we can establish very important key principles of human perception, but also about brain processing, neuronal processes, which I think are important then to develop also translations to clinical applications. For example, when we think of sensory loss or neurological or psychiatric diseases, which also pertain to perceptual problems, for example, but also when we more think about technical applications, for example, the design of food, or if we want to have sensing technical systems like robots, for example. And in order to follow the approach, I think one needs quite a lot of diverse interdisciplinary and also translational competencies in the sensing science, which I made some ideas about. And for the interdisciplinary part, I think we need some fundamental knowledge about cognitive neuroscience. That means that we know, for example, what perception actually is and the psychological perspective, what attention, but also means, because it's very closely interlinked, all attention and perceptual process, but also, for example, memory is important. And of course, one needs some solid background knowledge about the brain, about neuronal processes and functioning, of course, and definitely also some methodological skills about neuroimaging, but also neurostimulation, for example, transcranial magnetic stimulation. Further, I think one needs also knowledge for psychophysical experiments. That means also some physical or chemical knowledge, how to design actually stimuli. and also to design proper experiments which you are helping really to draw valid inferences on the processes which you want to investigate, but also some mathematical skills to do computational modeling and to precisely describe perception. And when especially also looking at neural data, neuroimaging data, for example, one needs also very much statistical knowledge, but also machine learning knowledge, because neuroimaging data sets are often very, very complex, and it has been established to use then machine learning algorithms to analyze this data. And yeah, I think overall you need, as a student or also as a researcher, you need quite diverse interdisciplinary competencies and skills to work in this field. And also when thinking about how to translate actually the fundamental principles then to applications, I think one needs, for example, market knowledge if you want to design products, but also clinical knowledge if you want to cure some disease or treat some disease, of course. And of course, you need also some knowledge about how to start a startup or building some business models and so on. And I think there are many potential educational formats where you can convey such knowledge, for example, on a small scale, like workshops like you actually do now, or lecture series or lab visits, or do joint real projects. I very like the idea of design thinking, that you put some students together and they try to work on a real-world problem. But on the larger scale, I think to transmit such interdisciplinary and translational knowledge, you need also like graduate schools or research centers, so that you build really a structure where we have a regular exchange of knowledge, actually. And at the FEU, we are currently building a research center for sensory sciences, where we exactly also want to implement such educational formats. Yeah, thanks for the opportunity and for listening, and also thanks for all the inspiring talks which really show how diverse and broad sensory sciences are.
Thank you for your presentation and also for your suggestion about the educational formats. So we have to move to the next speaker, who is Alberto Mazzoni from the Scuola Sant'Anna, and he works in is the head of the computational neuroengineering lab, and please, the floor is yours.
Thanks. So I hope you can hear me and see me well. So
yes.
Okay, so I'm not going to talk about sensors. I'm going to talk about sensory processing and infopathies, which is a word that I made up, but basically it means that we can study neuropathies as dysfunction in information processing. So my lab is the Computational Engineering Laboratory, and we do study information processing in the nervous system. We combine tools from computational neuroscience, like modeling and statistical analysis, as the previous speaker was saying, to develop applications for biomedical engineering and neurorobotics. In particular, we are interested in understanding the differences between the neural dynamics in healthy and pathological neural systems, and to develop neurostimulation therapies to tackle pathological neural dynamics. And we are not the only one having this approach. I think it's very interesting what in the introduction of this beautiful book Montgomery says, which is that all neurological and psychiatric disorders can be seen as misinformation related to the patterns of electrical activities in and among neurons. So all this is, to quote to the talk of Tosetti, this is a conceptual, not revolution maybe, but let's say a reform. So you can see all the symptoms and rephrase them in terms of misinformation, and this would be very helpful. For instance, I've been working for almost a decade together with Professor Micera and Professor Oddo on closed loop upper limb neuroprosthesis. And there is a lot of hardware involved, of course. So you have decoding of motor intention from the EMG analysis in the stump. Then you have a robotic hand that nicely grasps the objects, and some very excellent sensors that feel what are the properties of the objects. And then you have some electrodes that enable you to inject sensory patterns, if you want, into the nervous system of the patient. But the quest, my personal quest within this big project was what is the right algorithm to convert the sensor's output into the language of sensing of the nervous system. So across the years, we have done in the group several studies, and the nice thing to me was the interplay between neuroengineering and neuroscience. So the progresses in neuroscience helped us to build better artificial neural patterns to inject. And at the same time, our clinical tools with closed loop neuroprosthesis gave some confirmation or refined some way the knowledge of neuroscience on how peripheral information is processed. And so one thing we have learned in the end that we had to model in a very careful way the properties of mechanoreceptors at the periphery, and also to understand what is the interfaces that we are using to deliver this artificial sensor information. So in this case, all the neuroprosthesis work can be seen also as replacing some missing information. But this is not the only case. For instance, again with Professor Micera, we studied how diabetes type 2 could be a problem of misinformation, because the carotid body, who's a chemoreceptor, was chemoreceptors and is informing the sympathetic nervous system about the content of the blood, might trigger, if instead of giving the right information gives fake news about the content of insulin of the blood, can trigger some sympathetic overactivity that leads to diabetes type 2. So it's better to have no news than fake news, as we proved when we saw that both with the resection of the nerves from the going out from the carotid body, or better because it's more selective and reversible, with bilateral continuous stimulations, we were able to prevent metabolic dysfunction associated to a high-fat, high-sucrose diet in rodents. So neuroprosthesis replacing missing information. In this case, blocking misinformation, diabetes. We're also studying how, so a lot of neural disorders are affected by the visual cortex dynamics in the lab of Matteo Caleo at CNR in Pisa. For instance, migraine involves neural visual perception. As you know, you're afraid of bright lights when you have an headache, at least is my case. So we studied altered visual information processing in migraine, and we found, for instance, that the alteration wasn't in the firing rate, but in the frequency of the oscillations of the visual cortex, and this enabled us to develop a model that was able to capture what were the most relevant synaptic modification associated to migraine. So in this case, the altered visual information processing study was the key to understand the etiology of the disease. Last, again, together with Professor Micera, we are studying also information processing for motor disorders. In this case, we were studying in Parkinsonian patients, reach and grasp movement, and this C-score here is a coordination score and indicates a lack of coordination in the different, among the different joints when the patients were performing reach and grasp. And then we compared these kinematic data with the neural data acquired from deep brain electrodes in the subthalamic nucleus of the basal ganglia. And what we found was that while the mainstream theory said, so it matters, you have a problem if these oscillations are too strong, actually what we found was that the lack of coordination was associated to, again, to a loss of information carried by this oscillation, rather than them being strong or weak. So this is it, and I hope I convinced you that the multidisciplinary study of the processing of sensory information in the nervous system can shed a light on etiology of neuropathies and help develop therapies based on neural stimulation. But since here we are also dealing with interdisciplinarity, I would like to highlight in this last slide that this study needs to integrate neurophysiology, clinical neuroscience, computational neuroscience, and neuroengineering. And just to show that how we care in person about these multidisciplinarity practices, right now I have a joint lab with CNR, so rodents neurophysiology. On the other hand, if you want the clinical spectrum, I have a joint PhD with Würzburg Würzburg University Hospital, so interface between neuroengineering and neurology. And I also have a complex joint course with physics department of University of Pisa, so an interface between neuroengineering and complex systems. And I think we really need these at this moment to understand, not only to understand how the brain works, but also to fix it. Thanks.
Thank you. So fake news are inside, I mean, within our bodies, not only outside our bodies.
Exactly.
Okay, let's move to the last speaker of this session, Professor Angelo Recchia, who is full professor at the Scuola Sant'Anna, and sorry, Fabio Recchia, yes, he's full professor at the Scuola Sant'Anna, and he will talk about sensing in the heart, I think. So.
The cells in general. Can you hear me? Yeah, you
yes.
Can, right? Can you hear me? Yeah. Okay, so thank you very much, Tommaso, and thank you, Calogero, for inviting me to this high-tech symposium. So I will talk about water dispersible 3D nanoresonators for biological and medical applications. So we started this study years ago at this point to address an unmet need in biological and biomedical research. The need for intracellular nanosensors for the detection and quantification of biochemical and or biophysical change in vitro and especially in vivo. Speakers before me have already highlighted this particular need. And so if this is this is a schematic representation of the cell with a typical dimension, the typical diameter is between 10 and 100 micrometers. So a dream for cell biologists, but also for physiologists like me, would be to have free, harmless, nanoscopic intracellular probes which can enter the cell and detect there concentrations of metabolites, for example, such as glucose, fatty acids, lactic acid, or so on and so forth, or chemical and biophysical parameters like pH, PO2, PCO2. And again, this would be fantastic, but so far we don't have such a thing, especially in terms of quantification. So the way I was intrigued by this particular need, and years ago I had a collaboration with the Nestor laboratory of the Scuola Normale, and I asked two physicists there, Angelo Bifone and Alessandro Tredicucci, to put together our ideas. You know, physicists are very smart people, we know that. So they came up with an invention that was then patented. And also thanks to the collaboration of Vito Clerico, who was a PhD student in charge of this project at the time at Scuola Sant'Anna, and also other investigators at the Nest. Some of them are connected and are speakers in this symposium today. So what was the solution that they proposed? They proposed nanoresonators as nanosensors. This is the idea. So they proposed to fabricate a three-layer nanoresonator made of gold, aluminum oxide, and aluminum in this case, as you can see here. And I mean, they tell me, physicists tell me, these are physicists of condensed matter, that this type of parallelepiped can function as a nanoresonator, an L-SID circuit. As you can see here, it is in the nanoscale range. This is how they look like during fabrication, still attached to the base, to the matrix. And this is how they look like under electron microscopy when they are detached, like little bricks, I would say, right? And this is what we need. We need water dispersible free nanosensors. So how do they work? I mean, this is complex advanced physics, and I'm not at that level, but I will try my best, and physicists who are connected will forgive me. So the idea is this: if you have an infrared source and you that generates photons, if these photons don't have the wavelength, of, I hope that this goes, no, so this did not go, okay. If these don't have the wavelength of resonance of this nanoresonator, they will go through and they will be detected by an external detector. But if they do have the same wavelength of the resonance wavelength, their energy will be trapped in the nanoresonator because it will, in fact, elicit a resonance, and it will not be detected by the detector. So this is then detected as an energy absorbance at a particular wavelength, which is the resonance wavelength, and this depends on the shape and dimensions of the nanoresonator. In this case, it is 1.2 micrometers, very convenient for biological applications, in case we can discuss this later, why. But we can do better than that. So the previous wavelength may change if some molecules like long polymers, they function as nano antennas, molecular antennas, are connected, are bound to the nanoresonator. And here, this would lead to a change in the resonance wavelength, especially if this molecule interacts with the other molecules that we want to detect. So in other words, this shift will indicate the presence of the molecule or atom, whatever, that we need to detect. And not only that, but in theory, I mean, hopefully, will also be proportional to the amount of these molecules. So the solution that we propose here is the use of these nanoresonators. They are sharp-edged, but still probably very useful as nanoprobes that can enter the cell or even subcellular compartments like mitochondria and sense. Unfortunately, this technology, and it was again published years ago when it was still preliminary, remained only partially developed. The inventors took other paths. I mean, there was no possibility to continue. But in my opinion, this can still reveal a tremendous potential. In fact, there are still problems to solve: further reduction in size, homogeneous dispersion in water environment, functionalization with special molecules named Trojan horses for the penetration in cells, and specific functionalization that is molecule or biophysics specifics. To do that, we need complementary competencies and consequent training in alphabetical order, here cell biology, biochemistry, and physiology, but also chemistry, engineering, nanotechnology, and physics of the condensed matter. I will stop there.
Thank you very much. It's really a pity that this technique cannot be further developed.
Maybe it will be in the future, right?
So, Calogero, it's 1:10. What we are supposed to do now?
Thank you all for participating to the roundtable session of the morning. I think it was a very, very interesting debate, and let me say that it is clear there is a strong cluster of competencies in the area of sensing, meaning sensors and senses, applied to biomedical sciences and technologies. Also the, let's say, the colleagues that had some presentations in more transversal key enabling technologies, such as colleagues in the area of photonic sensors and so on, have clear possible applications in the field of biomedicine. So I think that this is something we can focus on for future initiatives. We don't have to decide now, but the discussion we had today can be foundational in launching some initiatives for the future under the umbrella of ELISA. I also see clearly that there are even internal possible sources of collaborations that were not activated yet, but maybe the workshop would activate. As an example, what Fabio Recchia has just shown could be of interest for the competencies of Stefano Palagi, just to make an example, because the micro nano robot that he mentioned could be applied to the research that Fabio mentioned. The workshop, I would conclude the morning session so that we can have time to eat, and we will continue in the afternoon session at 2 with a focus on applications, and in fact we will have a presentation by an industry representative that is, by the way, our partner company both at the Scuola. We have a framework agreement with ST Microelectronics, and the vice president of the competence center Artis 4.0. And after, there will be also Giovanni Comandè discussing legal and ethical issues in the remote sensing society, and Paolo Dario as a final keynote. After that, we will have a roundtable to conclude the day. In the roundtable in the afternoon, we hope to launch possible initiatives for strengthening the collaboration, the potential, also with the support of ELISA program. So let's see if we can move from the investigation and design phase up to the implementation. That will be the next step of ELISA. So if we can define together a common initiative, as an example, education initiatives, from weak initiative to stronger and more solid initiatives, and we will maybe discuss in the after. So I will stop the recording. Lorenzo, maybe you wanted to add or conclude?
No, just a big thank you to all the speakers, because it's a lot of problem and question are on the table now. Were put on the table. The entanglement of several disciplines. I agree with you. The entanglement of several disciplines in sensing, science, and technology area is evident, which largely justify this workshop and justify the call for a general reflection too on how the ELISA network should think and implement good practices in interdisciplinarity. So I'm very happy. And I think we can, I think there are some general issues to which the roundtable could pay attention in the afternoon session. First of all, what are the point of contact between engineering and non-engineering fields involved in sensing, science, and technology area? The main point of contact that there are a lot of point of contact evidence, but I think it would be useful to identify some point of the main point of contact to organize the next step of this common reflection, and how can these points of contact could be developed in an ELISA training program, for example. Could be a good topic to think about, because we should think about an ELISA training program dedicated, for example, to sensing, science, and technology. And what are the most appropriate tools, educational, scientific, to achieve this kind of disciplinary broadening, in order to meet the challenges identified today in this first part of the round table. So we have a lot of questions to discuss. I think could be useful to send to all participants a sort of a survey to integrate what is debated now in the round table and the workshop today. For example, the timing of introducing the interdisciplinary content into the curriculum. This is a big problem. A proposal for innovative teaching, proposal about bridging models for enhancing interdisciplinary knowledge in this area. There is a lot of problem and question that could be put in the table, but we haven't the time to do this now. But perhaps could be a good idea to prepare to organize a sort of survey for the participant and to have some outcome at the end of this workshop. So thank you very much for to all speakers of the morning, and good meal.
Thank you, Claudio. Thank you, Lorenzo.
Grazie, buon pranzo.
Ciao, grazie.
Grazie a tutti.
Grazie.